Well, welcome everybody to this webinar about Simcenter Culgi. And primarily, I will talk today about formulation development optimization with the Simcenter Culgi solution for the pharmaceutical industry. Quick agenda. I will give a short introduction of Simcenter Culgi and what you can do with it and how it fits in the larger Siemens portfolio. Then I will go into some case studies targeted to the pharmaceutical industry. And then I will end with some key benefits and Q&A session. Okay, so I guess this is an important slide to start off with. On the one hand, you're primarily developing your new chemistry in your labs at the R&D stage. And at some point, you need to bring that to the processing stage and then further on to the plant scale. And what we see when we look at software solutions that are out there in the market, both commercial and open source, there are quite a lot of tools to actually connect the processing to the engineering scale and the plant scale. So think about tools like you have with computational fluid dynamics or discrete element particles. That's all available, but much less so if you want to connect the chemistry to the processing stage. And with the Simcenter Culgi, we try to address some of those challenges. And so yeah, you see this big valley, and we try to cross that big valley using our solutions. So if you then place that into a more, let's say, model-based approach, you see on the left, you see the very small scales. So that's typically where you have some sort of quantum chemistry package. There are plenty out there that you can use. And then you slowly go into the molecular scale models where you have atoms interacting via simplified potentials. And then at some point you hit the coarse-grained models. Again, this is another scale-up where you basically take a certain number of atoms and put them together in larger particles. And then at some stage, you actually end up even with larger scale models where you don't necessarily have particles anymore, but maybe interacting fields. And then, of course, the idea is now that we want to connect it to the processing scale. And that, of course, is quite challenging because we need to connect those different scales. So both from the length perspective, but also from the time perspective. And what we envision is that with our Simcenter Culgi solution, we can try to tackle these aspects. Another way of looking at it is, well, you might ask yourself why you need computational chemistry in the first place. Well, traditionally, of course, when you go and set about trying to come up with a new product or to improve a product, you have some sort of experimental setup, some design space that you're interested in. You do your testing of your formulation. You analyze the results, and then you hopefully come up with a more optimized formulation for your actual application. However, what we try to do with our tools is to actually start off first doing design phase, where you can have a quite large data set of formulations that contain then all these different molecular species that are relevant for your application. You do some validation study using computational chemistry, and then you select your formulation and calculate certain properties, and thereby come up with an optimized formulation. Important, of course, is that during that, let's say, enriched digital experience, validation is, of course, very important. I will definitely address that as well. And validation typically means either some additional simulations that you need to do or experimental data that you have available. The one slide, well, actually two slides about the Simcenter Culgi solution. So it's actually a computational chemistry package. So it's not only targeted to the pharmaceutical industry, but it can help many different industries. We also have created different ways of working with the software. On the one hand, we have a so-called non-expert user interface, which is fully graphical, allowing you to drag and draw molecules, construct molecules on screen, build your simulation boxes, attach calculators and analyzers. So it's very much, let's say, anybody with a high school degree or has some experience with chemistry should be able to actually use that tool. Then we have a more advanced tool targeted to, let's say, a computational chemistry expert, which is a so-called scripting environment, which is still fully graphical, but allows to write very complex scripts. And the reason that we have that is because we see that many of the applications that our customers are interested in are, in the end, very complex in terms of the chemistry that goes into that. And then finally, we also have a means to connect to other software solutions, not only Siemens solutions, but any software solution that has a Python API. So we can then generate much larger workflows. So you can imagine that you start off with maybe a calculation at the quantum level, you get some properties from there, you import that into Culgi, calculate another set of properties at a higher scale, and then move that to even larger scale. So that's then fully connected workflows. And then at the list of properties, it's actually quite, I would say, vast. So you see here some properties that you can calculate with Simcenter Culgi. So it goes really all the way from solubility, miscibility aggregation, interfacial properties, rheological properties, phase transitions. I will say a little bit about that as well, but also more quantum mechanical based properties. So it's all available in the solution. And you also see here on the right some examples of the potential accuracy you can get with these models. So you see here, for example, melting boiling points of alkanes, very good agreement with that, or diffusion coefficients of small gas molecules going through a matrix. But we also have tools, for example, to calculate kinetic parameters of certain reactions that might occur in your systems. So in essence, a quite generic tool for computational chemistry. And the crux is really, I would say, in this slide. Of course, it's all very nice that you can run these so-called coarse-grained models, where you lump atoms together in larger particles, and thereby you can go to a larger length and time scales. But in the end, how accurate is that, and how useful is it for your application? So that's, I think, what it is all about. So what happened is that in the 90s of last century, Shell, actually the people, they were based in a Shell lab in Amsterdam, they developed the so-called dissipated particle dynamics method, which is a coarse-grained method to allow you to simulate liquids in let's say, in confined areas. And that was picked up actually by Unilever in the UK, and they developed a chemical awareness for that model. And that has from there taken off and has shown to be a quite important tool or tool set to actually allow you to go to these larger length and time scales that are typically relevant for many formulation problems. Important aspect, of course, is parameterization. So within the Simcenter Culgi solution, we have developed quite a lot of time and effort to actually come up with a means to calculate interaction parameters in a rigorous automated way. So within our protocol, within the software, we have tools to allow you to actually calculate those parameters on the fly based on certain theories. There is actually quite a lot of literature data available that you can go to. It's quite extensive, but in this webinar, I will only show basically the basic principles and not go into the details. The idea is that you start off with a fully atomistic model that's either available in your database or in our database. And we can also construct, of course, these models or import them from other software solutions. And from there, we do an automated fragmentation using Monte Carlo algorithms to actually cut up the molecule in these coarse-grained particles. Those coarse-grained particles also have associated with them a Cosmos service, which kind of tells you how good or how bad certain fragments will mix with each other. So it basically is a negative of the environment that is seen once you place those fragments in a conductive environment. So that means that if you, for example, have hydrophobic patches on your molecular surface, so you see, for example, in this triglycerides, you of course have these three aliphatic tails. those of course will be hydrophobic and will be, let's say, repellent for example, for water particles. But of course, the head bead, so that's the glycerol part, has oxygen in its structure. So that means that it's slightly, let's say, hydrophilic in nature. So we try to capture that in our model, really from the quantum chemistry level upwards. And then you end up with your coarse-grained model. A bit of background information of the type of potentials we're using in these type of modeling approaches. So on the one hand, of course, you can select a Lennard-Jones potential, which is quite standard for many classical force field-based simulations. So it has, of course, a very repulsive term, and it has, let's say, a well that basically tells you that at some point these particles will attract each other. But if they come too close together, there's a huge penalty. This works great, and it can be very, very insightful and very, very useful technology. The only real drawback is that it's rather slow in terms of amount of CPU time that you need to actually run a simulation. So basically, accurate but relatively slow. Then going forward into what I discussed a couple slides ago, about this dissipated particle dynamics developed by Hogenberg and Kummer from Shell. They developed this very simple approach where you have, let's say, only a repulsive potential, the Hogenberg and Kummer potential, that only has one parameter, so very, very simple to actually, get let's say, parameters out of for that. The only real drawback is that you can only simulate, let's say, liquids, because it's only repulsive in nature, so you cannot really simulate, let's say, phase transitions or multiple phases in one simulation. But there's the big advantage that it's extremely fast, so roughly 3 orders of magnitude faster than traditional, using the traditional Lennard-Jones potential. We have combined, let's say, information from both worlds. So we have our own Simcenter Culgi approach, which we call the cubic approach. It's slightly more, let's say, involved in terms of, let's say, the equations that go into it. So it requires a little bit of additional parameterization, but that has been fully, let's say, incorporated and optimized in the software. So that potential will allow you to simulate phase transitions if that's important for your application. And thereby, you still have, of course, the huge benefit of doing this fast equilibration. Okay, so you see here some examples of that, how that works in practice. So this for simple paraffins. So you see pentadecane, which is at room temperature liquid. And you see also the longer one, the hydrocosane, which is actually will melt at 40.5 degrees Celsius, so slightly above room temperature. And you see when you start your simulation in, let's say, amorphous state, and very quickly, this hydrocosane will actually start forming semi-crystalline domains appearing in the simulation. So that gives some confidence in the models. And you see here that we're applying this coarse-grained approach. So you see here on the top the actual all atom models, but then on top of that there is also the coarse-grained model that we use in our simulations. Another example, a little bit more advanced, is the solidification of oleogel formulations. So what we do here, we have sunflower oil, and we add glycerol monostearate to it. And then you see also that we can do, for example, a temperature gradient. So we started at high temperature, and at some point we start the cooling process very quickly. And you see also the immersion of these crystalline domains appearing in the simulation. And from there, we can, for example, calculate DSC curves to understand better the different phases that occur in the system. And thereby, we can also, for example, optimize the point where you see this, where you see the crystallization happening. And in this way, we can really follow or optimize the formulation until it's the right properties of for a given application. Okay, then short poll for you. So if you can fill this in, that would be great. I will just wait, let's say 10 seconds or something like that. So the question is, have you previously used digital tools or simulation software for drug formulation development or for computational chemistry in general? And if possible, could you name the tools that you're using? If you feel uncomfortable telling this, that's of course perfectly fine. And you can just skip this. I see the response coming in, that's great. All right, I'll do a couple more seconds here. Okay, I will just press next now. Okay, well, this may be this interesting, of course, information for Siemens, but maybe also for yourself. So I see about 25% using Schrödinger, 8% Material Studio, 8% OpenEye. Quite a lot of people are still using or are using open source tools. And also about half of you not using them at the moment. And also about 25% considering of using it. And if you then use these type of tools, do you use also some form of AI or machine learning at present in your research? Okay, I see that people are responding. That's great to see.
Cleaning and housekeeping is one area that we struggle with. What can be more mundane than this task? But did you know that the Zen practice treats cleaning as a form of meditation? Monks clean their temple, not just...
Sneha, I hear you, sorry. Okay, so yeah, quite a lot of you actually are like, almost 75% are using AI or machine learning tools. Great to see. And the rest not, okay. And maybe as a final question, I think, whether you see some relevance in these tools or just nice to have, or that you think, okay, this is really the future of materials modeling. And this is the last one. All right. Okay, let's move on. Okay, well, I think this is good to see, right? Nobody says it's not relevant, and then relevant is quite high, 60%, and good to have 40%. Okay, thanks for providing that input. I will now talk a little bit about the applications that we're working on. So let me just show the full screen. A little bit of background information, how we then apply this automated fragmentation and parameterization to biomolecules and proteins. well, again, we started with the atomistic representation. We have, as I explained, in the software a means to use a fragmentation algorithm using and then once we have the coarse-grained representation, then we basically have two choices. So we can use our Cosmo RS approach, where you have your surface, your Cosmo surface associated with the protein. And we use both a term that has, let's say, a free energy component and a volume component. And from there, we can calculate the interaction parameters that we need in our coarse-grained models like DPD. So you see here a small movie of that in the bottom. That's one approach. So that is, let's say, fully optimized. The alternative is to have, let's say, a more traditional approach using medicinal chemistry, where we basically say, okay, we have let's say, per amino acid is one particle in our coarse-grained model, and then we assign a certain type to that. So meaning that it can, for example, be hydrophobic in nature, hydrophilic in nature, neutral, charged, or has the capability of forming disulfide bonds, right? So we just summarize, we can have several approaches to actually do this parameterization scheme. And then big challenge, I guess not only for us, but for any type of software that you might want to use in this field, is so-called denaturation effect. That can happen, right? So these protein structures, they might not be stable depending on the environment or the temperature that you apply. So what we have done, we have basically combined some tools that are out in the literature. So on the one hand, we use DeepMind to actually come up with the right protein structure, or have at least an educated guess of that protein structure. Then we again apply this parameterization scheme where we fragment it. So we basically have our own mesoscopic coarse-grained model of the protein. And then on top of that, we can apply, let's say, a denaturation model. And I must say, this works quite well, but I would also have to say that it's still under development. And it really depends on the application itself, whether it's successful or not. So that's still quite a lot of hand-holding, and does not really work out of the box. But we do have some capabilities there that might help you in your research. And we know, of course, that depending on the environment, that it can be quite important. Okay, then this is immunoglobulin test case, basically, what we looked at. So quite a big protein antibody. We applied this fragmentation protocol, and using this medicinal chemistry approach, we generate interaction potentials for the different particles that are in that protein. And then from there, we actually do titration. So we basically start off with low pH, and then we go all the way to the high pH, so to calculate the isoelectric point. And that's a great measure to see whether, depending on the, let's say, the environment, whether we actually have a match with experimental data, because typically these isoelectric points are known experimentally. But that's a sort of a first validation step that we can do to qualify and assess the quality of our coarse-grained models. Once we have that, we can, of course, do studies of, for example, aggregation. So here you see an aggregation of these proteins in a solution, in a buffered solution. And what we do is we basically put those proteins at a certain distance from each other that they don't interact. Then we run the simulation. So this is a three-dimensional simulation. And we look at what happens over time. So we basically have a time trajectory. And what you see, for example, here is that this protein has, for certain buffers, a tendency to actually aggregate. So that's what you expect to see here. So we start off at, let's say, A, where you don't have any interaction, proteins don't have interaction. Then we go to point B, where you see indeed that over time, and you see also time axis in microsecond, you see there's a big tendency for those proteins to aggregate depending on the type of buffer that is used. And what you then also see is that we get semi-quantitative agreement between simulation results and experimental method dissociation constants, right? So you see here a table with the different types of buffers that we have added to the simulation. And then for certain buffers like citrate, succinate, and phosphate, they basically stick together, right? So they form a tight pair. And that's also what you see when you do the experimental measurements. Whereas for example, histidine and acetate, this is not the case. And that's also what we see in our simulation. So basically, they will not form, they will not aggregate. And what is also nice is because we have the underlying atomic structure, we can actually go back to that atomic structure and look at what points, what positions in the amino acid sequence they actually form this type, what are the amino acids that are responsible for that, aggregation effect, right? And of course, once you know that, there's of course possibility to redesign your antibody structure by mutating the specific amino acid sequence, provided of course that it does not interfere with the actual operation, the efficacy of your antibody. But at least gives some insight and some tools to actually start directing the actual development of the protein structure itself and start to change the protein structure itself. Another example of this, so again, we're doing buffer screening, but now for different temperatures. You can imagine that if you can store your vaccine, for example, at room temperature rather than at 5 degrees Celsius, that can be a huge plus in terms of cost, right? Because you don't need to ship it using all kinds of cooled environments. So again, similar type of approach, right? You see here now not 2 but 3 proteins, and now you basically follow again over time what happens to those proteins when they are in a solution. And you see here different types of excipients and different temperatures for a given pH. So if I summarize that, it could look like this, right? So you have your design of experiment, you have a set of buffers that you want to test, you have a set of excipients you want to test, and you might want to change the salt concentration and the temperature. So with that, yeah, you might think, okay, this can be quite costly to carry out in terms of raw materials. Well, I don't think that was relevant for your case because it's in the poll. But also, of course, you need to carry out those experiments, and it needs to be reproducible. So what we try to offer with our solution is to have a more cost-effective way of doing this virtually. Typically, every point that you need to calculate in this table takes about 8 hours on 4 CPUs. 4 cores. So that's not a big thing, right? You don't need a huge supercomputer for that. It's easy to monitor, right? You can look at the job status, and it should shorten your time to develop vaccines. And also the big plus is that you have now the actual all atom models that either work or don't work, right? So you have real insight of how your formulation is behaving at the atomistic level. Another example is when we apply shear in our simulation. So you see here, for example, a very, let's say, high protein formulation. So it has 10 grams per 10 millimeter protein, so quite high concentrations. And what you see here is that apparently at 145 degrees Celsius, there is still not a lot of going on unless you go to too high concentrations. So in this case, 40.40 grams per 100 milliliter. But it gets even worse when you go slightly higher in temperature. So you go only 5 degrees higher, and then you see a huge, somewhat dramatic increase in the viscosity. And this is triggered by the denaturation process that occurs, that the protein actually starts to denaturate. And what is nice to see also when you drill down into that, into the model and look at, for example, interaction energies between those proteins, you definitely also see that in the results, right? So you see here, typically not a lot of interaction at, let's say, low protein concentrations and below 145 degrees Celsius. But once you hit that denaturation temperature, it really jumps up, and that's exactly what we're also seeing from viscosity results. So this is the same type of approach, but then we apply additional external force to actually calculate the viscosity response. And this is how it then looks in practice. So you can have multiple customizable applications. In the end, it's not really required that you are a computational chemistry expert, although it will help. But these applications are set up in such a way that you just need to load your protein models or your biomolecules, give the concentrations and the weight fractions, and then the simulation will run. And on top of that, once you have finished your, let's say, your primary simulation, there are, of course, many applications to actually do the analysis of that simulation. So in that sense, it's readily available for anybody that has some notion of chemistry. Okay, so I think to summarize, Simcenter Culgi can help you to fast formulation design, where you can screen the chemical space for high performance formulations, to actually also perform virtual stress tests to drastically reduce your material consumption, hopefully prevent critical mistakes by having a fully digital thread, and also accelerate upscaling by answering questions that you might not have thought about without the access to a digital molecular model. So in the end, we try to offer something that satisfies all these conditions that you're looking for. So, for example, avoid protein aggregation, as I have shown. So, summarizing in one slide, so the key benefits of the Simcenter Culgi solution: you have a fully automated and customizable parameterization scheme that work for real proteins and biomolecules. You can do virtual screening at the micron length scale. You also have the possibility to map and backmap to all atom models and vice versa. We have a very open data structure allowing you to import, export information and data and files from other software to actually generate a seamless workflow. And also we have the possibility to generate these simplified graphical user interfaces for non-technical users. And with this, I would like to thank you for your attention. And I think there's still time for questions.
Yeah, thank you so much. We do have time to answer some questions here. So attendees, please type your questions into the Q&A Ask Questions widget. We did get a few here already. So I will start with those while we wait for some additional questions to come in. All right, let's see. So the first one that came in while you were presenting, it reads, what are Culgi's quantitative structure property relationship model development tools available? So what are the development tools that are available?
Yeah. So, so I did not go into that, but we have our own chemical informatics calculator that allows you I believe, to calculate up to 400 different descriptors. And from there, you can use all kinds of regression analysis.
Okay.
So it's actually quite, I would say, there's quite a lot of things you can try out there.
Okay, great. Thank you. Let's see. Can we simulate freezing and or drying, example, sublimation processes? Asking from a small lattice time scale perspective, and how long would it take for a given volume of simulation estimate?
Yeah, that's a great question. So we're also developing models for freeze thawing, which I think might be relevant here, judging from your question. So it depends a little bit on how fast or how slow that process is. But in essence, we should be able to also simulate that quite well, because we can introduce these so-called models that allow you to have multiple phases in one simulation.
Great, thank you. All right, our next one. Can you offer some comparison to other competitive tools or highlight Culgi's potential advantages?
Yes, that's from John, right? Yeah, so I tried to summarize that in one of the final slides. But I think what you can have with Simcenter Culgi is, like I explained, is an automated way to actually get the parameters that you need for your models. That's one part of it. And secondly, we also allow you to actually seamlessly go from one, let's say, one approach to the other approach. So if you, for example, start off with your fully atomistic model, that you can use your map to your coarse-grained model, do the equilibration there at the, let's say, the faster timescales, and then map it back again to the all ultimate models to do further property estimation, for example. But I think we have there a quite unique solution.
Great, thank you. All right, we have one more here. Again, attendees, you can keep those questions coming in through the Q&A Ask Questions window. We will get to as many of them as we can. If we don't get to your question, we can always follow up with you offline. So definitely keep putting those questions into the Q&A section. All right. Is the DeepMind approach to build structure of arbitrary molecules part of the package, or is that done offline? I'm trying to understand how integrated the process is.
Yeah, that's a great question as well. So it's actually it's not integrated, right? So we basically it's like an offline tool. So we use yeah. So it's not integrated, but it's not very difficult to do, right? So once you know how that tool works, it's relatively straightforward to actually come up with a structure and then load that into the Culgi software.
Okay,
but
great.
it's a great question. Yeah.
All right. I'll give everyone another minute or so to get any more questions in that you might have. All right, we've got another one. Let's see here. How well, oh, sorry, another two. How well does the rheological property estimation model work for complex formulations? From Leihing, number 8 there.
Yeah, it's oh yeah, that's from Lai, right? Yeah.
Lai,
okay.
yeah.
So how well does the rheological property estimation model work for complex form? Yeah, that's a difficult question to answer. It really depends on the property that that you're interested in, and on the amount of computational power that you have. So some properties are relatively let's say straightforward to calculate. Others are much harder to do. So yeah, it's maybe a little bit more. You probably were hoping for more better answer, but again, it will depend on the property.
All right, thank you. All right, we've got a few more here. Let's see. Do you think ML can help in coarse graining?
Yeah. So I mean, coarse graining is as building those models. I think there is a certain need for machine learning. So you can well, there are two approaches, right? So on the one hand, you can say, okay, I'll just take my full complexity of my molecular model and then use that as an input for the coarse graining. That will take let's say a certain amount of time. I think maybe hours. So, and that's usually okay. But of course, if you're doing, let's say, a very big databases, let's say of tens of thousands of molecules, then it's very advantageous to use some sort of machine learning tool where you can reuse information that you have already acquired in previous steps. And in fact, we have tried that in certain applications, and that works well. But I mean, I must say, for most of the applications that we worked on, it was not really necessary. So, yeah, the actual coarse graining took much less time than running, for example, the simulations.
All right, great, thank you.
I hope that answered the question.
The next question, from your experience on the topic of speed versus accuracy, is there a break even or an optimum? For an accurate rough cut simulation, a baseline would help. Question number 10.
Again, a very difficult question. I mean, I don't know. It depends again in what industry you're in. So based on my experience, then usually accuracy is more important than running longer simulations. So typically, yeah, I would say that if you apply computational chemistry in your research and you want to make an impact, then getting accurate results is more important than running it very quickly. Although, of course, there is, of course, a trade-off, right? So you can imagine if you need to wait half a year for a result, that's not feasible, right? Maybe for a PhD or for an academic project, but not for yeah, let's say if you're in a commercial research organization. So we try to get results within days, and at most within weeks, right? But yeah, you can imagine if you want to have a very accurate result, and that might take 2 weeks of calculation time. It still might be useful to do that because of the accuracy then what you will end up with. But it's a great question.
Okay, thank you.
It is.
And then the next one that came in, what are input parameters into the model? Do we need to know protein structure?
Yeah. So okay, that's a good question. Yeah, so we need to know the protein structure indeed. So we like I said, if it's not known from, let's say, from a protein database readily available, then we can apply DeepMind or Rosetta to actually come up with the protein structure. But yeah, in the end, it's a requirement, I would say, because we need to calculate the charge, the charge density. So yeah, it's definitely a requirement.
Okay, great. Well, thank you. I do see a couple more here. A couple people are asking if there are any demo versions or trial periods available for testing the software. Yeah,
so this is Israel, right? We're actually working on that. So at the moment, I don't think so, but let's say within hopefully not too long time, we will have a freely available demo version that you can try out.
Okay. And we do have a contact us information widget, so definitely be sure to click on that if you would like to get some more information. But I don't see any more questions here at the moment. I do see a few thank yous coming through, so I'll pass those on behalf of our attendees. So thank you, Jean-William, for presenting and for answering all of these questions today. Thank you very much.
Thank you, Sarah.
And it looks like our questions queue is still clear, so I think we can go ahead and wrap up our Q&A. I would like to let you know if you think of any other questions once we close, you can email me directly at sarah.alspot@siemens.com and I can forward you or point you in the right direction. Just a reminder, this session is being recorded. You will receive an email with a link to view the follow-up within a couple of hours. So keep an eye out for that so you can, of course, watch that again and share that with your colleagues as you see fit. If you have a project immediately in mind, this is a phone number that will take you to a list of, this is a QR code rather, that will take you to a list of phone numbers. So you can find the appropriate phone number for your location and call in and get connected. And again, you should also see the contact us widget, I believe, I'm not sure which way to my left, but you can click on that and that will take you to a website where you can contact us as well. All right, well, this concludes our webinar. Thank you so much for being here, everyone. We hope you have a great rest of your day, and we'll see you soon. Bye.